Hello, everybody. So today we're going to keep going down the path of trying to describe mathematically how the lag operon is regulated. And just as a reminder, go back here, we have this situation where the E. coli can tell when there's glucose, when there's no glucose, and when there's lactose around. And only in that condition does it produce the machinery necessary to metabolize lactose. And it does so through a combination of activation, that we will talk about probably later, and repression. So you see this repressor binds to the DNA in the absence of lactose. And as a result, RNA polymerase cannot bind to the promoter and cannot make LAC-Z, and other proteins are responsible for lactose metabolism. And in the previous lecture, we started by coming up with a model for the constitutive promoter. We're trying to see how what are the features of that dictate the expression of a constitutive promoter in terms of parameters such as, for example, the binding energy of the repressor to the RNA polymerase to the promoter, or the number of RNA polymerase molecules. And we introduced the statistical mechanics protocol. And if you remember, one of the key first step of this protocol was to think of the different states that the system can be found in. So in the case of the constitutive promoter, we had an empty promoter or a promoter bound by RNA polymerase. And after some math, by using Boltzmann recipe, you can come up with their corresponding statistical weights. So these weights look a little bit different from what we did in the last lecture, but they're gonna become clear once we start doing some math together. But just to give you a sense, these weights already allow you to identify the different knobs that you could tune both experimentally and theoretically to test your model. Because you can see, for example, the number of RNA polymerase molecules showing up, the binding energy between the RNA polymerase and the promoter. And actually, this is the delta epsilon that we talked about last time, which is actually the difference between the binding energy of polymerase to the non-specific to the specific site minus the binding energy to the non-specific site. And also, there's a contribution from the number of non-specific binding sites from the reservoir, which basically tells us where is RNA polymerase hanging out when it's not bound specifically to the promoter. And you can calculate the probability of finding RNA polymerase bound to the promoter, the probability of the polymerase bound state, by dividing the weight of this state by the sum of all possible weights the system can be found in. And we played a little bit with plotting the probability of finding RNA polymerase bound to the promoter as function of the number of RNA polymerase molecules by playing with different axes, linear axis, log axis. And here I'm showing you one example of p-bound on a log linear axis, where I'm showing you p-bound for two different binding energies. One is for the lag promoter, okay, in red. And the other one is for the T7A1 promoter, which is a promoter from the T7 bacteriophage that infects E. coli. And the T7 has a very interesting lifestyle, which is that it first injects its DNA, and one of the first promoters that comes in is the A1 promoter. And RNA polymerase binds to it and starts transcribing along the DNA, and that basically pulls the DNA into the bacterium. So you would imagine that in order to do this, you want a really strong promoter. And indeed, you can see that the probability of finding RNA polymerase bound to the promoter is very close to 1. In contrast, for the lag promoter shown in red, you want to, in principle, you would want to have some range to go up with activation and to go down with repression. And you can see that for the constitutive lag promoter, the probability of finding the probability of finding RNA polymerase bound to the promoter is, 1 in 1,000 or so. Okay, so but this is all good, but what I want to do now is to go back here and think about not just what the polymerase is doing when there is no transcription factor, but to think about what happens when you have a repressor binding site. Okay, all right. So let's get started with that, and then we'll go back to talking a little bit about how you test this experiment. So the first thing we're going to do is to come up with a cartoon model of simple repression. So this is a cartoon model of simple repression. Okay. And what is this cartoon model? Well, I'm going to say that I have my little promoter region. Okay, so it's a promoter. And then I'm going to have a little repressor binding site, so repressor site. But what I'm going to say is that when this repressor is bound, Okay, so this is the repressor. It doesn't let RNA polymerase, RNAP, doesn't let RNA polymerase bind to the DNA. So it's excluding RNA polymerase from the promoter. Okay, so we want to turn this into math. We want to be able to predict, for example, the level of gene expression as a function of the number of repressors in the cell, as a function of the interaction energy between the repressor and the DNA. And to do that, we're gonna follow the StatMech protocol just like we did for the constitutive promoter. So follow the statistical mechanics protocol. And what are the steps? So the first thing is to identify the state the system can be found in. Right? So let's do it here. So I'm going to have here states. So what can happen to this system? So let's just draw our little promoter with a repressor binding site. And I claim that three things can happen here. First of all, the promoter can be empty. Okay. But all the things that can happen is that it can be bound by RNA polymerase. Check it out. So RNA polymerase could be bound here. Or it can be bound by the repressor. And note that I'm not including a state where the repressor and the polymerase are bound at the same time. So basically, again, when repressor, they exclude each other. There's like steric hindrance, if you wanna give it a more specific name. Okay. All right. So this is the model. Well, these are the states that we're going to consider. Okay. So what is the next thing that we need to do is we need to think of the the energies, right? So assign energies to each state. Okay, so let's do energy. Okay, so in this case, where the promoter is empty, I still have pRNA polymerase and R repressors, right? So this is let's do it like so. This is RNAP number, and this here is the repressor number. And just like we did for the constitutive promoter, each one of these guys is going to be bound non-specifically somewhere. So there's going to be an non-specific binding energy of polymers to the DNA, and also the R repressors that are in the non-specific reservoir are going to be bound with a non-specific binding energy of epsilon and NSRD. Okay. Now, if I have one polymerase that came out of the reservoir to bind specifically, I'm going to have P-1 epsilon PD non-specific plus R epsilon RD non-specific plus now P epsilon specific PD, right? Sorry, not P. There you go, right? So this is the one polymerase, one RNAP bound to promoter. Okay. And now let's consider the case of the repressor. Well, in this case, what am I going to have? I'm going to have P RNA polymerase is bound specifically, R-1 RNA polymerases, repressors bound non-specifically, plus epsilon specific of repressor to the DNA. Okay, so this is one repressor bound specifically. Okay, so far so good. All right, so what's next in the protocol? Well, the next part of the protocol, if you remember, is to figure out, enumerate the number of ways each state can be realized. Basically, we need to figure out what the multiplicity is. All right, so I'm going to do multiplicity. Okay, and last time we said that the problem of RNA polymerase hanging out in the non-specific background is the same problem as having like 3 or 4 people coming into a 30,000 seat stadium, right? Like the 3 or 4 people, you can assign them randomly to different seats, and it's very unlikely that they're going to see each other. Okay, now if you on top of it add 3 people that are with the RNA polymerase t-shirt and 3 more people with the repressor t-shirt, it's also going to be pretty unlikely that they're going to see each other. Okay, so what I mean by that is that the multiplicity is going to be just the same that we had for the case of the constitutive promoter, but now accounting for the fact that I have both polymerase molecules and repressor molecules, meaning I'm going to have NNS to the R plus P, right? Remember, because we have the first polymerase has NNS ways of binding, the second polymerase has NNS minus 1, but it's kind of like NNS ways of binding, and so forth. And then I need to remember that I had these factors that accounted for the fact that the RNA polymerase molecules are indistinguishable, so this is a P factorial, and also that repressor molecules are indistinguishable, this is an R factorial. Okay, all right, what about the next one? Well, here I lost one of the polymerase, so this is going to be NNS R plus P minus 1 divided by P minus 1 factorial divided by R factorial. And the final one, move this up a little bit just to make some space. And the final multiplicity is going to be NNS R minus 1 plus P divided by P factorial R minus 1 factorial. Okay? And finally, I'm ready to compute the Boltzmann weights. So the last step of the protocol was to compute the Boltzmann weights. Okay, so I'm gonna start a new line for this. Copy this. So I'm going to do Boltzmann weight. Okay, and what do? I multiply I do the multiplicity times e to the minus beta the energy. Okay, so because I'm kind of lazy, I'm going to grab the multiplicities like this, copy, and now I'm going to do the energy. So this is e to the minus beta P epsilon non-specific polymerase to the DNA plus R epsilon non-specific repressor to the DNA. This is e to the minus beta P minus 1 epsilon non-specific polymerase to the DNA plus epsilon specific polymerase to the DNA plus R epsilon non-specific repressor to the DNA. And finally, this is e to the minus beta P epsilon non-specific polymers to the DNA plus R minus 1 epsilon non-specific precursor to the DNA plus epsilon, epsilon specific precursor to the DNA. Okay, so we're done. We can now calculate the probability of finding the polymerase bound to the DNA. Okay, so step 5. Calculate probability of state we're interested in by dividing its weight by the sum of all possible. For possible weights. Okay, so let's do that. So P bound, it's going to be a little bit nasty, but you're going to see it simplifies quite nicely like we did in the case of the constitutive promoter. So P bound is the weight corresponding to RNA polymers being bound, right? So I'm going to move it here. It's going to go here. And now divided by the sum of all these weights, right? So it's kind of nasty. But you're going to see in a second. It's going to get a lot nicer. Do plus like this. Okay, so now what I'm going to do is do the same trick I did last time, which is multiply by 1. Okay, what do I mean by that? Check it out. I'm going to multiply by 1 by doing in a very specific way, which I'm going to say is I'm going to multiply by b factorial r factorial divided by nns to the r plus p times e to the minus beta p epsilon ns pd, sorry, e to the plus beta plus r epsilon non-specific rt. Okay, and I'm going to have the same thing below. Okay, p factorial r factorial nns r plus p e to the beta p epsilon pd non-specific plus r epsilon rd non-specific. Okay, why did I do this? Because note that if I multiply, like the terms basically is the inverse of this, right? So this turns into a 1. Okay, so what else happens here? Let's see. Here, maybe what I'll do is do I want to do each one of these independently? Well, let's multiply this term with this term here. Okay, so what's going to happen here? Let's see. It's kind of nasty. Okay. Let me just do it up here then. How do I want to do this? I'm just trying to be lazy, and I shouldn't be lazy. So, all right, let me do it on this side here. Okay, check it out. Here we have r plus p minus 1. Here we have r plus p, which means that here these two guys are going to go away. Okay, we also have, and we'll do it like this, r and r here, right? So these two cancel out. sorry, this cancelled out already. This and this cancels out. Okay. Also remember that p factorial divided by here, p factorial divided by p minus 1 factorial is just p, right? So I'm gonna get rid of this guy and this, and instead I'm gonna have p. Okay. Now let's look at the exponential. I'm gonna have e to the r, e to the beta r epsilon, check it out, epsilon and SRD, and this is the same as this one. So this is good because I can get rid of it. Okay, and note that here I have p minus 1 or minus p minus 1 because there's a minus here, and p. Okay, which means that I can get rid of this whole thing. I get rid of p. Okay, so the numerator of this thing is going to look like, let's see, the NNS that I have there goes to the denominator. I had a p that came from here, okay, and then I have an e to the minus beta epsilon, no, epsilon specific PD minus epsilon non-specific PD, and that's it. That's everything I have left. Okay, below I'm gonna have the 1 of this term, this term here, right? So 1 plus. Now, this term here is the same as the one we had in the numerator. So it's going to be p NNS e to the minus beta epsilon PD s minus epsilon PD NS. Okay, and if you go through the math for the last term, what you're going to see is that it's going to give you r over NNS e to the minus beta epsilon specific RD minus epsilon non-specific RD. Okay, so we can define a few interesting things. For example, we can say that this is delta epsilon PD, the difference between RNA polymers being bound specifically and non-specifically to the DNA. And this we're going to call delta epsilon RD, the same type of difference, but now in the context of the repressor, the difference between the energy of the repressor being bound specifically minus the repressor being bound non-specifically. Okay, so in short form, this can be written then as P over NNS e to the minus beta delta epsilon PD divided by 1 plus P over NNS e to the minus beta delta epsilon PD plus R over NNS e to the minus beta delta epsilon RD. Okay? Okay, so there are a few interesting things here. Like, note that I can always, as statistical weights, I can always, if I multiply all the weights by the same thing, they're still statistical weights, okay? And what this tells us is that maybe there is such a thing as renormalized weights that are a little more intuitive. So what I mean by this, we just uncovered the renormalized statistical weights. What do I mean by this? Check it out. Let's borrow. I'm going to borrow my states drawing from here because I don't want to draw it again. Bring down. Okay. Now here I'm going to write the renormalized weights. Okay. So in the renormalized weight language, the first state has a weight of 1, right? The first state has a weight of 1. The second state has a weight of p and ns e to the minus beta delta epsilon rd. And the recursive bound state has r over ns e to the minus beta delta epsilon, sorry, this was pd. Polymerase to the DNA, repressor to the DNA. So, when I think of these StatMech models, at this point, I don't go through this whole math, okay? Because I'm so used to them that I know how to write the renormalized weight. And if you have some practice with it, if you start playing with it, you'll realize that there's a really nice structure here, okay? All right, so now I want to do one more thing. I want to try to figure out, what I want to try to simplify this expression for p-bound. Maybe I won't do that yet. Actually, I'm not going to do that yet. Okay, so what do we have? We just calculated p-bound, right? And now the question is like, we have a prediction. The probability of finding RNA polymerase bound to the promoter is given by this formula, which has information about the number of polymerase, their binding energy to the DNA, the number of repressors, the binding energy to the DNA. In principle, all variables that you could tune, you could tune experimentally, that you might have some access to experimentally. Okay, all right. So let's see. What do I want to say? But it's tricky to measure absolute binding probabilities. You probably are familiar with example with ChIP-seq, but there are many things that you might worry about with ChIP-seq, where even though the claim is that it measures binding, I haven't seen a convincing calibration between an actual measure on binding and a signal from ChIP-seq, which, as a reminder, is the result is just counting how many DNA sequences you get in a certain region, right? That ultimately you can relate to binding, maybe in some way, but it's not even clear that it's linear or in any way. So they're not really, despite how much we think about binding and how much we use binding in our language when thinking about the genomic control, for example, there are not really good ways to measure occupancy in vivo. Okay, and so what did we decide? There are a few tricks that you can do, though. Okay, so remember that the idea was that the mRNA in steady state, right, was equal to R over gamma times p bound. Okay, and in this case, it's p bound that is a function of p and of r. Okay, so remember that in steady state, okay, so but absolute occupancies are hard to measure. Okay, what if, but at the same time, it's easy, yet it's easy to measure gene expression. Okay, like you can measure RNA relatively easily, you can measure protein levels relatively easily. Okay, so, but obviously if I calculate p bound, right, that's not enough to predict the RNA levels. I need to know R and gamma, and that can get a little bit tricky because I might not know R and gamma for whatever system I'm doing. But instead, one cool thing that it's easy to measure gene expression. Since we don't know R or gamma, we calculate a fold change. So what do I mean by this? Check it out. So I'm going to call the fold change gene expression. I'm going to say this is the mRNA steady state when in the presence of P and R divided by the mRNA steady state when I have polymerases, but when I have no repressors. Okay, imagine that I can do an experiment where I have repressors, I measure the number of RNAs, and I do another experiment where I got rid of the repressors, and then I measure the RNA, and I just take the ratio. Now, the cool thing is that, check it out, this is R over gamma times P bound of PR divided by R over gamma is P bound of P R equals 0, okay? And the nice thing is that now R and gamma disappear, like they cancel out. So now I have P bound of PR divided by P bound PR equals 0, which is a fold change in promoter occupancy. Okay, so my point is that we just connected something I can measure with something I can calculate. Okay, and this is really powerful. Okay, so now let's actually calculate what this looks like. Okay, and I'm gonna make this I'm gonna find some abbreviations here. Okay, just because I don't want to be writing that much. Okay, in shorthand, I'm gonna say this is 1, this is little p, and this is little r. Okay, why do I say that? Because then the fold change, and now I'm going to use the fold change in promoter occupancy or the fold change in gene expression interchangeably, right? Is P bound in the presence of polymerase and repressor. So that's P over 1 plus P plus R, right? And this is all a bit smaller. So divided by P over 1 plus P, right? Because R, little r, is this term here, and in the absence of repressor, the term is 0. Okay. All right. So what does this look like? Well, this is what P over 1 plus P plus R times 1 plus P divided by P, which once again looks nice because this cancels out, and I get 1 plus P, 1 plus P plus R. Okay. We're almost ready. We're almost done with this, but we're going to make one more assumption. One more. Or actually, instead of an assumption, let's examine the relative values of P and R. Okay, so P, remember, is big P over NNS e to the minus beta delta epsilon PD. Okay, and what are these saying? This is around 5,000. This we know that is 5 times 10 to the 6, and this is about for the lag promoter is about minus 3 kBT. Okay, let's do it also for R. Little r is R over NNS e to the minus beta delta epsilon RD. You know, there's only 10 black repressors in the cell. It's not too many. But they bind at a much higher energy. They can bind up to like minus 20 keV. Okay, so now let's do the math for these 6. I guess I can do it. What do I do? I'm running out of screens here. So maybe I'll do it here. Let's do it. Let's bring up. Let's see. Check it out. It's all there. MCB website. Okay, so I'm going to do 5,000 divided by 5 times 10 to the 6 times e to the 3, because that's minus 3 kT. Okay, and that's, I'm going to write it down here, 0.02. Okay, so this is about 0.02. Okay, now let's do the next one. I'm going to do 10 divided by 5 times 10 to the 6. So you would think, you know, this is going to be super small. But what happens with the exponential when you do e to the 20? You get 970. Okay, so whereas on the top this was 0.02, this is 970. What I'm telling you is that from this we learn that R is much larger than P. Okay, and also check it out, and also that P is much smaller than 1. Okay, so these are the 2 things that we learn. But that means then that the fold change, that I had up here, right? Under this so-called weak promoter approximation, right? 1 plus P, if P is much smaller than 1, that's just 1. And if P is much smaller than R, in the denominator, we get 1 plus R. Okay, so this is the fold change. And by weak promoter, I mean a lag promoter, right? No, I don't mean the T7A1 promoter. So this formula is not necessarily true for all promoters, but it seems to be true for the promoters that are, you know, under regulation in the, you know, under significant regulation in E. coli. Okay, so we're done. Okay, I can also write this fold change. Maybe we'll go back to the non-shorthand form as 1 over 1 plus R over NNS e to the minus beta delta epsilon RD. Okay, and sometimes people call this the repression, 1 over the repression. Okay. All right. So why am I excited about this? Because once again, I showed you that this fold change is a quantity you can calculate and you can measure, and we managed to simplify it into a very compact model with only 2 free parameters: the number of repressors and the binding energy of the repressor to the DNA. Okay. So let me just recap a little bit. Oops, what's happening here? You know, we used the statistical mechanics protocol to come up with the corresponding, the Boltzmann weights, or the renormalized weights corresponding to each one of these states. Okay, and here I have this extra column, if you remember from the previous lecture, where we're saying that, you know, the polymerase, the promoter empty state and the repressor occupied state don't produce any RNAs, but the polymerase bound state does produce some RNA. Okay, and basically, if this was the model in the constitutive promoter, what is happening when you have the repressor is that you get an extra state, and ignore the fact that you have 2 heads and you have a factor of 2 here. But basically, what's happening is that you have your binding energy here, your number of repressors, and the result is that the repressor is basically draining probability from the polymerase bound state. So if the rate of production, or the number of RNAs produced as a function of time, look like this in the absence of repressor is just slowing down the production. Okay. And as I showed you, when we calculate the fold change in promoter occupancy, we get this really nice simplified expression that features just the number of repressor molecules and the binding site strength. Okay, and I hinted at the fact that this is something that I worked on during my PhD, and I started thinking of this from the theory perspective, and I was super excited to have this um, these predictions. And then I thought, okay, I'm just going to go talk to some experimentalists and see whether they would be game for testing those predictions experimentally. And what happened is that nobody was interested in testing these predictions. Okay, so we had to turn ourselves into experimentalists. Okay, but despite how excited I am about this simple model, okay, that we just introduced, as you know, reality is more complex. Reality doesn't look like the simple repression motif shown here, where you have the repressor binding to a unique site. In the lac operon, you have some inducer that is basically related to the lactose concentration that comes in, binds to the repressor and makes it unbind from the DNA. There are pumps that pump this inducer. The inducer can also be transported passively through the membrane. The repressor, as I hinted at, has multiple binding sites. It can bind to multiple sites simultaneously, looping the DNA in between. And these are all sorts of complications that are interesting, but clearly they would make the model way more complex. Okay, so the first thing that we had to do is to think about ways of bending the lac operon, making it simpler so we can track it theoretically. So what do? We got rid of the inducer. We got rid of the inducer pump. We got rid of the DNA looping, and we created a synthetic lac operon with only one binding site. And sure enough, this is not the simple repression motif. Is not really what you find in nature. But if you can understand the simple motif, I claim that you have very little hope in understanding much more complex regulatory architectures. Okay, and we had an advantage, which is that this guy Benno Miller-Hill in the 90s did this amazing work showing that the simple repression architecture works, that you can change the binding affinity by modulating the DNA binding sequence. Okay, so people knew three binding affinities, three binding sites, or 1 or 2 or 3. That you can measure the repression as a function of two different numbers of repressors, and that repression is quantifiable. So, I was once at a talk by Sidney Werner, and one of the things he said is, you either want to be 6 months ahead of everybody or 30 years behind. And we were really definitely in the mode of being 30 years behind from everybody, because basically what we said is, let's revisit these experiments and let's try to contrast them with a theoretical model. Okay, so how do you measure this repression or this fold change in gene expression? Well, the way Miller-Hill did it is by actually looking at the enzyme that can catalyze lactose, and instead of giving lactose to the cells, he gave them a substrate that, when cleaved, becomes yellow. So then you can measure the rate of yellow production in cells that don't have the repressor, so they will make yellow at a higher rate, and in cells that have the repressor, you can see it here, so they will make yellow at a lower rate, and then you take the ratio of those two rates. Okay. How do you measure number of repressors? Well, one way that you can do it is by doing immunostaining, by basically doing dot plots. You can see here little dot plots where you just actually pipette in a little bit of lysate from cells, and then you flow in antibodies and use phosphorylated peroxidase, and you measure luminescence, and you have a standard where you add known amounts of your repressor, and you can use that to calibrate how many repressors you have in your bacteria. So, but the result is that now we can measure the fold change enzymatically and the number of repressors using immunostaining. Okay, and so now we have tools to compare theory and experiment. There's other versions of testing of fold change, and another version is to instead of looking at the enzymatic activity of LAC-Z, the enzyme that catabolizes lactose, to rewire the cellular decision to, instead of LAC-Z, produce GFP, such that now you can look at the fluorescence of cells that don't have the repressor. You can see these cells under the microscope that are really fluorescent. Cells that have the repressor, you can see that these are darker. You can measure the fluorescence of each one of these guys and get a single cell distribution and calculate the ratio of the means. So an alternative way to test the model is measuring fold change by fluorescence, and you can even measure the number of repressors by fluorescence as well. And basically, you're going to do this during discussion over the next few weeks, and this is going to culminate in a really cool homework problem. What you're going to do is you're going to learn how to analyze fluorescence from bacteria that have different binding sites for the repressor and different binding energies, different binding sites will represent different number of repressors, we're going to give you a set of pictures of bacteria where we took data in the phase contrast channel and in the fluorescence channel. Okay, you're going to find where the bacteria are by performing segmentation over the phase contrast channel. Okay, then you're going to overlay the segmentation with the fluorescence channel in order to calculate the fluorescence per cell. And you're going to be able to use this by measuring, for example, the fluorescence from cells that have the repressor, cells that don't have the repressor, to calculate the fold change as a function of the repressor number. Okay, so you put this all together. What are the sorts of predictions that you can test? Okay, and this is the type of conclusion that you're going to get to after you do the analysis. So, first of all, what are the knobs that you could think of tuning experimentally and theoretically in order to test our model? Well, we talked about the number of repressors, we talked about the binding energy. You could also think of moving the binding site and also changing the number of promoters. It's not the same to have 100 repressors on one promoter than to have 100 promoters on one repressor. Imagine if you put the gene on a plasmid, for example. So, but let's focus for now on modulating the number of repressors and the binding energy. Okay, what I'm showing you here is the level of the fold change in gene expression for a given number of repressors. Okay, where each color corresponds to a different binding affinity, to a different binding site. You see, OID is blue, is the one that gives you that lowest, the highest, the biggest fold change, right? Like the most deviation from 1. O3 is the weakest guy, that is the closest to 1. Okay, so these are measurements that give you, again, fold change in gene expression, number of repressors. So I know the number of repressors, I know fold change. I can fit the data in order to obtain an in vivo binding energy for each one of these binding sites. And the fit is fantastic. But of course, I'm joking, because all I showed you is that I can fit a 1-parameter model to 1 data. But the cool thing is that there are no more free parameters here. If I now tune the number of repressors, if I move along the number of repressors, the data better fall on top of these curves. And if it doesn't, it means I did, one of my assumptions isn't right. Okay? So we did that over almost 2 orders of magnitude. And what we showed is that to a large extent, we can precisely predict output levels of gene expression from knowledge of the DNA sequence and the concentration of the repressor. Just like you can predict the output of the output current of an electronic circuit as a function of the input voltage from knowledge of the circuit diagram. Okay, but before we finish, I gave you the speech about how we were bending the lag operand to make it simpler. So what happens now if we try to make things a little more complex? Can we use what we learned in the simpler scenario to gain predictive power about more complex situations? And one interesting knob to tune is the number of copies of your gene. You could imagine, everything I showed you so far has to do with a reporter integrated in a single site in the bacterial genome. But you could imagine having the reporters on plasmids, you could imagine having them on multiple sites on the genome, or you could imagine even having your reporter site on the genome in a single site, but having a bunch of competitor DNA that has a binding site for the repressor but no gene. Okay? And I'm not going to go through the math here, though. I can show you. I can refer you to some interesting papers where you can do the math, and it's super interesting StatMech calculations. But basically, once you go through the math, what you can show is that the fold change is given by this expression here, where you can see that we still have the number of repressors, we have the binding energy of the repressor to the DNA, and we have a new parameter that is the number of competitors. But the cool thing is that you can tell the number of competitors through some other measurement, like doing qPCR, doing measuring DNA content. There are independent ways of measuring the number of competitor binding sites. What I'm telling you is that we could then predict the fold change in gene expression as a function of the number of repressors, not only as a function of the in terms of the binding energy, but also in terms of the number of repressors. And these are the predictions that we made. And the point is that all these predictions existed before we took the data. Okay? And then what we did is we took the data. And what we showed now is that to a very large extent, even in this more complex situation, we can predict input-output functions for simple repression without having to invoke any free parameters, because everything I used were the in vivo binding energies that I obtained from simple repression and knowledge of the number of promoters. Okay. Not only that, all these curves look very different. They don't look like they stem from the same model necessarily. But this is because we're not plotting this plot with the right x-axis. So if you plot instead of the number of recursives, you plot a fugacity, which is basically an effective number of recursives that accounts for the competition between between the specific and the competing binding sites. You can show that all these curves, all these data points collapse into a master curve. And this is a very big deal in physics. When you can show that all your experiments that look very different collapse onto one master curve, it means that you found the natural variable of the system, the variable that actually matters for the system. Not to anthropomorphize too much, but the variable that the actual variable that the cell cares about. Okay, so the cool thing is that people sometimes say, well, there's no way you can be predictive about biology in the same way that you can be in physics. And every time they tell me that, I show them this example, and I tell them, look, I don't know, the one case that we tried for 10 years, it worked. Okay, and my point is that this is one step of the pyramid. Like I showed you what happens when we try to predict what happens when you have one gene copy. Okay, we can use that to predict what happens when you have multiple gene copies. There's also another layer that we could explore if people are interested that has to do with noise and transcriptional bursting. You can use that to predict then what happens when you add the complexity of inducer and when you add, for example, the complexity of changing different domains of the recursor due to evolution. And the point is that each step of this pyramid, every time we climb one step, we use the parameters that we learned in the previous step. And that makes the inference of new parameters in the next steps much simpler. So it's not like as I make it more complex, I need to know more and more new parameters, because I depend on the parameters that I learned in the first iterations. The number of new parameters with every iteration as I increase regulatory complexity remains manageable. Okay. And today I showed you an example of simple repression, okay, of what happens when you have a little repressor molecule that binds to the DNA and excludes RNA polymerase from the promoter. Okay, but this is one of many motifs, regulatory motifs, that you can model and predict using statistical mechanics. You can do simple activation. You can do activation with a helper molecule. You can do repressors interacting with each other, activators interacting with each other. There's all sorts of cool things that you can do, and we're just starting to scratch the surface of the types of models, of the types of ways that we can engage in this theory experiment dialogue to try to reach a predictive understanding of how the number, placement, and affinity of transcription factor binding sites in regulatory regions dictates gene regulatory input-output functions. All right. Well, this is it for now. I guess in the next lecture, maybe what I'll do is we'll think a little bit about cooperativity and how you make even sharp input-output functions, how you encode for genetic switches. Okay. Bye.